Hello there, ladies and gentlemen. My name's David T. Kramer. This is Sociology, and today we are talking about the scientific method. What is science? How do we define science? What kinds of evidence does a scientist look for? And the question that I know is on everybody's mind right now, what are the 7 steps in the scientific method? Keep in mind, sociology studies social reality. And social reality is different from physical reality. Humans make up social reality, and we humans possess consciousness, while physical phenomena, like gravity, do not. While we humans construct our social reality, each one of us has free will, and each one of us may self-select beliefs, values, norms, and roles, or we might even innovate and create new practices. How cool is that? I'm so glad I'm not gravity. What is science? Science is a systematic pursuit of knowledge using empirical evidence. Things that can be seen, smelled, tasted, touched, and heard. Not only by the scientists, but by others as well. We recognize that we humans are connected to our physical and social realities by our sensory organs. Science is published and open to criticism. We can hold scientists accountable. And we find well over 100 peer-reviewed journals that publish sociological research. Science is ethical. No Dr. Evils, baby. Honesty is imperative. Honesty with subjects, methods, conclusions, and reporting. Do no harm. Do not aggress upon another person or another person's property. Here we are. What are the 7 steps in the scientific method? Step number 1, select a problem, something that deserves to be studied. Step number 2, review the literature. Number 3, develop a hypothesis. Number 4, choose a research design. Step number 5, collect the data. Step number 6, analyze that data. And number 7, state the conclusions as well as limitations. Okay, something that I think is a problem. I've had friends, colleagues, people in my community die from heart disease. And the Centers for Disease Control report that heart disease is the #1 killer of Americans. I think heart disease deserves to be studied. And I'm wondering, are there any social variables? Is there anything about our social reality that's impacting heart disease? There is a sub-area of sociology called social epidemiology, and social epidemiology studies the relationship between various social variables like social class, nationality, culture, gender, occupation, and disease. Perhaps there's something about culture that has an effect upon the development of heart disease. Step number 2, review the literature. Well, I notice when I look at the World Health Organization that they collected data in the 1990s, and they looked at the percentage of calories from unrefined plant foods in various nations, various cultures, various social realities around the world. And they also looked at the percentage of deaths from heart disease and cancer. Look at Laos. The standard Laotian diet has over 90% of its calories. That's the green bar here. Over 90% of its calories coming from unrefined plant foods. And fewer than 10% of the deaths in Laos are from heart disease and cancer. But look on the left-hand side at Hungary. Oh my God, 90% of the deaths from heart disease and cancer, but fewer than 10% of their calories are coming from unrefined plant foods. What we have here is a correlation, and it's an inverse correlation. As unrefined plant food increases, disease decreases. But being good scientists like we are, and critical thinkers like we are, we know that correlation does not prove cause and effect. There has to be a mechanism there, right? Correlation does not prove cause and effect. There might be some other variable, something other than cultural diet impacting these killer diseases. However, as we continue our literature review, we come across the migrant studies, and the migrant studies show that when individuals move from a plant food culture like Southeast Asia to the USA and they eat the standard American diet, their incidence of heart disease and cancer go up as well. They didn't change their genes, they changed their diet. And our diet today, the standard American diet today, is even worse than it was in 1990. At least from the standpoint of eating whole fruits, whole vegetables, whole legumes, whole grains. Today, only 7% of our diet is coming from those whole plant foods. 51% of what we eat is coming from refined and processed foods, 42% from dairy and other animal products. Well, here's the all-American meal, minus the soda. Take that potato, take the skin off of it, deeply fry it in oil. Take that wheat, polish off all the fiber, throw in some high fructose corn syrup, preservatives, and various other chemicals, and bake up a bun. And then we take a cow, we feed that cow full of corn, antibiotics, and growth hormones, grind that cow up, and then we make a patty, and that patty, 64% of the calories in that patty come from fat. Here's a hypothesis. A single meal from the standard American diet will increase blood lipids, that is fat in the blood, that is triglycerides and cholesterol, and will inhibit blood flow. In other words, inhibit the blood vessel from dilating so that the blood flows well. What is a hypothesis? Hypothesis is an educated prediction. Why is it educated? Because we did step number 2. We reviewed the literature. A hypothesis is an educated prediction about the relationship between 2 or more variables. A variable is something that can be observed. Remember, see, smell, taste, touch, and hear, and that changes. A hypothesis will have both an independent variable and a dependent variable. The independent variable is the cause, and the dependent variable is the effect. The independent variable in my hypothesis, hamburger and fries. Dependent variables, blood lipids and blood flow. A single meal from the standard American diet will increase blood lipids and decrease blood flow. Pretty cool hypothesis, right? Well, now we need to choose a research design. Step number 4, choose a research design. There are several things that are available, quite a few actually. We could do an experiment in the lab, in the field. We might find a natural experiment where we have a treatment group and a control group, not because the scientists set it up that way, but it just happened that way. Or we could do a survey. Telephone, mail, online. Or we may find some archival data already collected. We could look at that. Observation. Become a participant in a group and observe people. We could do a case study. I think for this hypothesis, the experiment is the best approach. Step number 5, collect the data. And when you do an experiment, you have to pre-test your variables. So we want to pre-test our blood lipids and our blood flow. We want to measure the fat in the blood, and we want to measure how well the blood vessels are dilating, how well they are allowing that blood to flow, how well the endothelium is producing that nitric oxide that makes the blood vessel dilate and allows that blood to flow. Then we want to manipulate the independent variable for our treatment group, but not for the control group. So, in our experiment, let's feed the treatment group meat, white bread, fried potatoes, standard American meal, right? And let's feed our control group unrefined plant foods. Ooh, how about some fresh spinach leaves? How about a baked potato? No butter, because remember, butter is an animal product. Some strawberries and an apple. Oh my God, an apple has so much soluble and insoluble fiber and escorts those lipids out the intestine. Okay, then we'll do a post-test of our variables. We'll measure our blood lipids and our blood flow. How are we going to measure the blood flow? Well, we can use the brachial artery tourniquet test that was developed by Robert Vocal of the University of Maryland. He used an ultrasound probe over the brachial artery to see how well the brachial artery is dilating and how well the blood is flowing. Ouch! Collect some blood. Spin that blood up in the centrifuge. Now we're analyzing the data, right? We've collected our data. Now we're analyzing the data. Spin that blood up in the centrifuge. It will separate the red blood cells from the plasma. Oh my God, look at the plasma. Before the standard American meal, it's clear. After the standard American meal, you can see the fat in the plasma. Of course, we will want to actually measure the milligrams per deciliter of triglycerides and cholesterol, but you can actually physically see it before you analyze it further. Well, fortunately for us, this experiment has been done many times. Robert Vogel from University of Maryland has done it, and Robert Vogel reports that 2 hours after the American meal, triglycerides increased from 94 milligrams per deciliter of blood to 147 milligrams per deciliter of blood. Dilation of blood vessels was hindered up to 4 hours, and some researchers find up to 6 hours after this standard American meal, the blood vessels don't want to dilate. They're just not working right. Look at the subjects consuming plant foods. They maintain healthy blood vessel dilation, and the lipids did not increase. Wow. So, correlation does not prove cause and effect, but it turns out we have a lot of mechanisms that have been identified. We've talked about just one today. There have been mechanisms identified in terms of both heart disease and cancer. There is an inverse correlation. As unrefined plant food increases, heart disease decreases. And yes, using the scientific method, we can identify cause and effect relationships. We can identify mechanisms by which increasing unrefined plant foods decreases the likelihood of getting heart disease. Humans make up social reality. Sociology studies social reality. Culture is part of our social reality. The food that we eat, how we produce our food, how we prepare our food, which foods we eat are all part of our social reality. And yes, social reality is different from physical reality. We human beings possess consciousness, while physical phenomena like blood lipids do not. While humans construct our social reality, each one of us has free will, and we may reject the beliefs, values, and norms and roles found in our social reality. What does that mean? That means that just because you and I are socialized in the United States, just because you and I grow up in the USA, and just because we live in the USA, does not mean we have to eat like an American. I'm so glad I'm not gravity. I'm not a blood lipid. I'm so glad I'm a human being. Well, thank you very much for your participation today. My name is David T. Kramer. This is Sociology, and today we've talked about the 7 steps in the scientific method.